Good afternoon, everyone. I'm going to make one argument over the. Is this sound too loud? Okay, it kind of sounds too loud here. Yeah, over the next 60 minutes, and thank you, Diane, for giving me an extra long speaker slot. I know, yeah. Okay, so I have a very short version, so I'll give you the ending right now. In April, late April, FDA kind of initiated the building of real-time clinical trials, which basically is the back end of drug development. It's a great advance, but the point is that no one has built the front end, which is identifying which trial actually is even worth running. And I want to argue that we do have the machinery here, which is I2B2 and all these federated networks that we've talked. And so this is the short version, and let me show you what I mean. All right, so this is what happened. On 28th of April, FDA came out with the announcement that they're doing a first proof of concept, real-time, what they call real-time trials. They're underway, where basically safety and efficacy of signals were being reported to the agency as they were occurring. And the premise behind it was that the process of drug development that takes over 10 years should not run on signals that arrive years later. And a couple other things that the agency also announced was they're basically handing AI the work of dose selection, recruitment, and as well as the go or no-go calls. So here is the assumption that this initiative has. And the assumption, the underlying assumption that's hiding underneath all of it is that every one of those tasks needs a trial that already exists with an effect size, a population, and an endpoint, which is a big hypothesis. And this is the one thing a decade of clinical AI still cannot produce. So if you look at the work of AI in drug development, you see that most of the advancement has happened in phase 1 when the question is it safe? Is the molecule safe? Phase 2, which tries to figure out whether actually a drug works or it helps the sick patients, it drops to 40%. And phase 3, which is still kind of like first readouts are coming, the overall the success rate still is really failure rate sits at about 90%. So there's not much advancement in real throughout the drug development. So AI basically solved a third of the problem, which is which molecule, which is arguably the easiest part. But the second phase, the 2/3, are kind of untouched, which is really the question of who are the patients and what are the basically our hypotheses that are worth doing the trials on. All right, so here is the what I called where things break. As you all know, drug development is really brutal because it takes thousands of compounds that one of them maybe yields to a medicine. As we discussed, 90% fail rate. And the thing that kills most of trials, not the chemistry as we discussed, is one of the major bottlenecks is recruitment and the struggle to find the right patient at the right time. So as I mentioned, AI, if you want to be honest, AI did wins the, AI wins are really at the upstream task of finding the molecule, which is kind of not that expensive. The expensive really part is the late phases where you have to do, where drugs actually lie and most of the failure happens. So 2 questions here. The rest of this talk is around that. One is that what does it take to find the right patient and walk those right patients into the trial, a trial that is already knowing the right hypothesis. And okay, so I talked about the bottleneck, and I said that the bottleneck is not the molecule. It is finding the right patient for the right hypothesis before even the trial starts. So stay with that for a second. If we look at the second part of this question, the hypothesis. So this picture kind of encapsulates the current pipeline where the hypothesis already exists, yet there's no way to produce it. So it comes from the picture that data arrives fragmented and siloed across institutions, while someone assembles a cohort through different procedures, and then you get a small and potentially biased insight that lands years too late. Look at the bridge and notice that it never reaches the other side. This is the translation gap. And here is the line I want you to carry today. Real-time trials, as I said, are only as good as hypothesis that enters them. Okay, so I need to introduce this term because this one word, inference, holds the rest of this entire talk. So inference asks whether something changes an outcome by how much and in whom, here's the part that people miss. A randomized clinical trial and an observational study are both inference, and they differ only in how the data are mined. So in contrast to inference is prediction, and it's where it tells you what will happen to a patient. So inference tells you what changes that outcome and for whom, and this is exactly where drug discovery basically entirely lies. What changes for exactly which patient? Okay, so I want to argue, this is not a new argument. This is an argument that's been around prediction, where a machine learning model became more prevalent in computer science, whereas, more standard statistics. So there is two paradigms here. One paradigms, they basically kind of read the same clinical data, but they reward opposite instincts. Prediction wants one function that works as the bedside. So when different sites have different underlying population, it basically treats those differences as nuisances that have to be erased. Inference, on the other hand, wants an estimate that holds across population, and it asks how consistent that estimate is. So when sites have different underlying population or different findings, that becomes the entire signal. And then consistency across different sites becomes basically what the signal is in inference. How am I doing? Am I okay? So this basically single table holds the entire argument. So there's two columns. These are opposite projects rather than better and worse, basically opposite versions of the same thing. So prediction, as I said, learns a function and scores itself on those metrics such as AUROC and ships a model for individual patients, whereas in inference, we are estimating an effect and a score and are more concerned about replication and whether the hypothesis can be generated from it. So the decisive basically role here is the heterogeneity. In prediction, this is a liability. Heterogeneity in population is a liability, as we talked about, that weights have to be recalibrated across different sites. While in inference, the heterogeneity itself is the evidence. And there are studies that have shown that no loss function can reconcile populations that are genuinely different in characteristics. All right. But then what is inference? Is it too complicated? Is it a really concept? And the answer is that It is not actually. So what you guys have all built, all these federated networks, I2B2 as as a tool to identify patients and cohorts and phenotypes, networks like ENACT, the way that work together. These are all pieces of that machinery for inference, for distributed inference. We have built it, but the shiny thing, the AI has kind of set it a little back. The point is that the engine is already here. We just call it the query tool. So a query tool is basically an engine to distributed inference. And the logic basically is not new, as I said. In 1965, Bradford Hill basically built up that. He called consistency across population one of the strongest signs of causation. And the move that matters is that a finding from one institution might be nothing more than local confounder, but the same finding, if you find it across 50 institutions, cannot be a local confounder, because that confounder would have to live everywhere in the same direction and the same size. So a 50-sided study is not simply a bigger study. The agreement or maybe even divergence between the results that you get from different sides becomes the evidence. And I'll show you an example of what I exactly mean by this. So that's the argument that inference is the right paradigm, and it's been running for over a decade, as long as these federated networks have also been going on. Now, the obvious question is, if the paradigm is old, what does AI actually change? And so the AI doesn't change the paradigm. It offers resolution. It raises the granularity at which we can actually make inference and make an operational inference. And that new resolution is what makes what I call precision phenotyping possible, which is the second part. Okay, what is precision phenotyping? What I mean, so Griffin already went through phenotyping. There's been a lot of conversations. I did a whole presentation last year about what precision phenotyping is. And it basically, in single terms, is that rule-based phenotyping or small size computed phenotypes are basically prediction problems, right? So they're trying to predict one score for one patient for having that phenotype or not. Whereas if we look at in precision phenotyping, we are basically trying to come up with a much bigger model with a lot of different parameters that can bend into or mesh into different individual patients and provide the required definition for that patient. So one example, like think of AI enabling precision phenotyping like an LLM. What is the difference between an LLM, a foundation model? It has so many parameters. It can, if you give it a text, it can rewrite it in Shakespeare language, or it can turn it into a Mary Oliver poem, right? So it can do all of that. There's so many parameters in that, whereas some of them may not be useful, but we are basically thinking about the same thing. Having precision phenotyping algorithm in a way that it can basically optimize for patients, for each patient. And what happens in most of these things, as happened in AI, is that most of precision phenotyping, what I'm talking about, is based on attention. And attention is kind of embedded in time. The same way that LLMs work, it is embedded in time. So let's step back with me to the EHR that all of us hold dearly in our hearts. Health records, in my opinion, are the most exhaustive footprint of human morbidity and that we've ever assembled. So there is a lot of information in there. It captures the depth in health records, the longitudinal depth, captures transitions that no clinic or no trial would ever see together. If they're read in order, it can reveal the architecture of disease and the way one condition sets up the next. Um, so we took 435,000 sequences or ordered connections of electronic medical record data and distilled them to 118,000 real transitions that were created through these silver standard models enabled with LLMs and tested those against UK biobank genetics. And we found strongest aids were enriched for shared genetic architecture, which the record genuinely knows the order in which the disease moves. So there is ways to actually extract really meaningful things. And this is a really, this is an example I usually do, which basically disease is not a photograph, it's a trajectory of a patient. And if this is what Griffin covered. If you look at a diagnosis code, for example, in the medical records, it's like a memory loss diagnosis code on its own is 41% predictive of Alzheimer's. But if you put that diagnosis code next to another thing, for example, memory loss, memory loss is 57%, I think. And when it's followed by donepezil, it climbs to 72%. The same kind of connections between these sequences can make same increase in boost in the signal. We looked at the data on that 435 thousands of sequences in EHR data, where we are actually able to identify 5 recurrent, basically subtypes or archetypes of mechanistic pathways that you can see in the EHR data. We call them acute cascades, escalation chains, branching trajectories, feedback loops, human-mediated syndromes, and there is a lot of examples for each of them that you can find. But let's go to our flagship, basically, work that explains that is another use for that sequence data. So we implemented a definition of long COVID 2 years ago, and it basically lies on a lot of sequences of data, the same 435,000, and each of them can indicate a diagnosis of exclusion. Here is basically the definition for long COVID. We were able to implement it. We published a study in 2024. We also published a study about 2 weeks ago in JAMA Network Open, where we were able to actually distribute it across 4 sites, thanks to people like Giazi, Tian, who implemented, and Michelle Morris, who is involved in all of the good projects. So we found that long COVID can be in 16% of patients. About 90% of them are conditions that are chronic. But most importantly, as I was talking about the signal that we get and consistency in signal, we find some of the consistent things that we found that is not fading. The prevalence is continuing to grow, but there is also divergence in the signals. For example, in New England, thyroid predominant and endocrine manifestations were most of the top conditions that came up in Texas and South California. It was prediabetes. Same in Western Pennsylvania, 40 hospitals, and then you see rising quarterly prevalence. So these are the things that we found that you could never find from an averaged, federated, in a way that coefficients get weighted out. You can never find those kind of insights. All right, let's pause here, and because this is the human reason any of this matters. Our phenotyping rewards clean, common, single disease patients. So if you are thinking about rule-based and the older versions of computed phenotypes, we are basically rewarding those type of patients who have those single disease conditions because those are easy to name, a lot of time with the code, but the patients that are hardest to define are usually the ones that carry the most unmet need. multimorbidities, rare diseases, atypical courses of disease. They satisfy no clean eligibility rules, and that's why they never actually make it into real trials. No therapy ever gets tested for them. And this is the case for a lot of long COVID patients, for example. And long COVID is just one clear case in our own work that people are still, after years of the COVID pandemic, people are still trying to find cure, and they are still complaining that there is no cure. Now comes the pivot, and that I've been trying to, so I've been trying to sell basically precision phenotyping as recruitment and the right patient, which is basically finding the right patient at the right time. This is true, but it's a bad undersell of the machinery, because the same phenotypes that identify patient also can surface signals that are worth testing. So when you create these precision phenotype, develop precision phenotype, you create precision research cohort, you can actually test a lot of hypotheses. You can test drug repurposing hypotheses in a lot of subpopulation and things that are not usually possible to do. All right, going to the last part of the presentation. So there are replicated signals already that I'm going to show some of them. But here's the crux of everything. How does a signal become a hypothesis sharp enough to power a real trial? This is the basically handoff that the FDA's plan depends on, and never actually was built. So as I mentioned, what actually gets handed up is not the finding, but a structure object with 4 parts. There is an effect size and a defined subpopulation and a condition. It appeared under direction of mechanism. So when a trial is bad, making the trial real time isn't going to make it better. It's just, it's not going to fix the fully rescue false hypothesis. Every part of these elements in clinical trials basically feed directly into trial design, and a trial built on that already knows what it is hunting before it starts. So these are 4 studies that 2 of them are ours. One, we did a study on Paxlovid on long COVID and the monoclonal antibodies. And so in the Paxlovid study, we showed that unlike previous studies that at the time were showing there's no effect on long COVID, we are actually able to show that in the general population, yes, there may not be a sizable effect, but in particular subgroups of population, specifically in people who have GI sequelae, there is a 37% drop in the risk. And so this type of question, we did a similar type of study. So if you wanted to answer this type of question through regular clinical trial pathway, first of all, you would never find enough people with subgroups of GI long COVID, and then it would take a long time and a lot of dollars to be able to test these things. And we were able to do it in weeks. Same type of studies we do with we did with monoclonal antibodies. We used a cohort of roughly 118,000 patients, and we showed that both antibody therapy and vaccination track with lower post-COVID burden. On monoclonal specifically, there are a lot of interest, but there is again not enough sample size. Now we have curated these cohorts across 4 institutions. We can test them. We can test these hypotheses in any of those institutions. Another problem is that where does the idea of the monoclonals can have an effect come from? This is another thing that we can test and build this complete translational arc. So this is well, basically the whole thing closes. And this is the entire picture of what I showed at the beginning. So as I mentioned, if you read it from left to right, the left is where we began with fragmented data, manual cohorts, and years lost. The middle is the missing piece, which is a distributed inference, running locally and replicated and consistent. And the right is what falls out of that distributed inference mechanism, which is a trial-ready hypothesis with its effect size and population and mechanism already specified. So there is already studies, for example, there's a GPL-1 study that was run recently in the VA data. They looked at they basically looked at the effect across 170, I think, outcomes in VA data, including cognitive endpoints, and nobody was looking for it. So this is how they found out they were able to actually, it replicated in independent networks and then entered the pipeline, clinical trial pipeline as EVOKE in early Alzheimer's, which is a real world signal that was carried end to end into registered trial. And this is what the entire story is that I'm talking about. There is also, in addition to the science, there is also a financial twin for promoting distributed inference and distributed networks. If you look at the cost spent on all of us, about $2.5 to $3 billion is the cumulative appropriations to all of us, which basically the number of patients that was recruited in his database. If you look at the total cost is $5,000 per patient, EHR-linked participant. There was budget cost, same thing, centralized ways they're trying to happen in entry C, but not really fully trial-ready kind of hypothesis come out of that. Now we have experience with PCORNet, we have ENAC, we have a lot of experience with distributed networks that can actually deliver the same thing, not only much better, but also much cheaper. All right, so let me just finish with this part on the where I started. So FDA's initiative compresses the back end, and that is genuinely really a work that is worth doing. But it can never compress the front end and where hypotheses are born and chosen. A trial that arrives already knowing its endpoint and its effect and its population from replicated evidence, it's a different animal from one that learns all of that over years, no matter how fast you watch the data. I believe that we can, using precision phenotyping, creating precision research cohort and its federated inference networks, we can make the end-to-end trial pipeline much faster and much more reliable and actually address patients that need drugs that cannot find the treatments. Finally, I just want to thank you, my collaborators and funders, especially Giazi and Michelle, who really made that long COVID study happen. Thank you.
All right, thank you, Hossein. We have time for probably one or two questions if we have any in the audience.
Hossein, it is so nice to hear your large language mind again. Yeah. So let's see. Talk about that. What do we have to do to get these edge cases identified and get back over the bridge so that they're addressable with mechanistic reasoning and inference into like what could really target what's going on? So, let's say with the GI stuff, did you have accompanying stool samples that showed like viral particles in there that could have then, served as a like a companion diagnostic for focusing in on them, and that they would be addressable with the Paxlovid stuff, for example? Okay. Those are two really different questions, actually.
Okay. Yeah. First of all, your first part about my large language mind, is it that my mind is large or my... Thank you. Well, I think for your question, first of all, what can we do like to make these things happen? So what we are basically building is we are building an attention mechanism through these, large number of sequences, and that's how we implemented long COVID definition. We are able to identify. Now we have the cohort, so um, there are hypotheses that we can generate, including drug hypothesis, or like what are the risk factors that we can actually, because we have such large groups of subtypes of long COVID, now I think vet labs can actually test some of these hypotheses, and it's
I think the bridge is really adequate for them to understand
Absolutely.
what would be the reasoning space within that subgroup.
Yes, I think this is where the reach of electronic health records basically goes. You cannot really tap into.
Yeah, I don't agree, because I think that there's stuff that is just out of what's in record space that never gets there. You're not going to have it. You have to do mechanistic reasoning.
Absolutely,
this,
yes.
this phenotype patterning.
Yes, that's what I'm actually talking about. I'm saying that you can generate hypotheses, but for mechanistic reasoning, you have to actually have molecular data and then go do. And this is what vet labs can do. So it doesn't close the entire. We are just trying to prepare the right hypothesis that is worth probably further analysis in trials, and then have the population. The rest is.
Okay, but Recover was $1 billion. How much? And the clinical trials that were attempted were pretty lame, and that the reach to patients was virtually nil. Tell me about how that makes sense. Do you have a sort of similar
Yes. So my argument, I don't have any arguments against Recover. This is, when Recover was started,
we needed a bridge. What's the?
Yeah, we needed a bridge, but we now know that we needed a bridge, right? At the time, something has to be created. So I think now we know that we needed a bridge. But if next pandemic happens, do we already know that we need it? So I think this is a course of discovery that, and you just look at the investment and Recover. It looks large, but it's not large compared to every other investment that's happened in this country. So I think it's kind of needed. It was a needed effort, and I'm glad that it's moving on, and it's contributing to some of the urgency of where to look at for other people who are doing the same type of research with smaller amounts of money.
Very interesting framework. Something I think about a lot. I do wonder, though, if also the same framework could be used to refine endpoints. So I know in clinical trials, endpoints have to be predetermined, right? But also using your framework, you could also start to see, well, this one endpoint is not generalizing across these populations, but perhaps there's this other thing that is changing consistently across the populations. So do you think there's an opportunity to sort of like use the same framework to say, okay, we can refine our endpoints such that more drugs cross the finish line? Because what informs biomarker discovery is sometimes pretty much, it's not always well done. Biomarker discovery is hard. So this is potentially one way to do that. And the other thing is, if it's not refining the biomarker, it might be refining the indication. So I give you a drug and I think I'm curing cancer, but maybe I'm not curing cancer. I'm instead curing diabetes in people who have another comorbidity or something. And so you can on the fly also do kind of like this kind of repurposing of the drug in clinical trials as they move forward, potentially. So I just wanted to put this out there as maybe question/idea to think about in terms of how you can capitalize on sort of this kind of framework that you're thinking about.
Yeah, that's a really good suggestion about endpoints, because there's already enough endpoints that still there are trials that has to happen, but there are endpoints, as you mentioned, that are hard to define, like long COVID, because first the endpoint, sometimes the endpoint is the phenotype. Sometimes the phenotype is what predicts the endpoint. And one of the areas of active research for us is we're looking at endpoints that can measure recovery and aging, you know, and longevity. And you can find it, you know, hundreds of endpoints, but how many of them can you measure with the electronic medical records, and how many of them do you have enough consistent data? And then when you build the endpoint, you can then build the phenotype of who are at risk of different endpoints. So I think it's basically which part is your phenotype definition that has to be, you know, it can be applied to all of that.